Here is on.
Ah.
Sim, vou precisar. I'm going to present yourself. Bom dia a todos. Faz sentido usar o micro? Faz, se calhar faz. Faz. Somos muitos. Bom dia. Ai, tá. Mas está ligado? Está ligado. Ok. Bom dia, obrigada a todos e obrigada por darem também aqui à oftalmologia a oportunidade de trazer um convidado e estarmos aqui um bocadinho juntos, que eu acho que também espelha aquilo que depois é o nosso dia-a-dia neste hospital, em que a oftalmologia e a reumatologia estão muito próximas e é bom lembrar-nos e termos estas sessões de informação. Eu vou apresentar o Colin em português para ser um bocadinho mais emotiva. O Colin Chu é investigador principal na UCL, atualmente é especialista em uveites no Moorfields, que é o maior hospital de oftalmologia da Europa e provavelmente um dos maiores do mundo. Tem o segundo, tivemos ontem a falar, a segunda maior coorte de doentes com uveíte do mundo. O primeiro é nos Estados Unidos, na Flórida, e o segundo é em Londres. E é uma pessoa que me ensinou muito, que eu conheci em Bristol em 2016. Provavelmente eu hoje faço uveites porque conheci o Colin e mais 3 ou 4 pessoas em Bristol e, portanto, isso foi muito importante formativamente para mim e pessoalmente também. Ele tem uma investigação extensa do ponto de vista de técnicas de imagem avançadas em tecidos e, sobretudo, a inflamação ocular, e tem um papel muito importante em trazer a investigação básica e translação para a clínica. Tem atualmente vários estudantes de doutoramento em Londres, com projetos muito impressionantes, e acho que vão gostar muito de o ouvir.
This one.
Just spoke the truth about you.
Thank you, Ines. I hope so. If someone can give me feedback later. I'll just switch that. Does that need to go off? So thank you so much, Ines, and thank you all and the department for inviting me here. It's a real pleasure to talk about my work, and it's really great to have a mixed audience. Just for my benefit, how many people work in rheumatology, are rheumatologists in the room? The majority. And how many are my fellow ophthalmologists? Minority. So that's good, because I'm going to start nice and basic with a reminder on the eye. And what I hope to do today is really just give you an overview of some of the work my lab's starting a journey on. So really, I started my lab at UCL and at Moorfields Eye Hospital only about 2 years ago, after doing various postdocs in the US and working in the University of Bristol, where I met Nesh. And hopefully what I've really focused on is not going too deep into lots of data, because I knew it would be a mixed audience, but really to hopefully show the ophthalmologists some exciting work we're doing to better understand the biology and pathology of the eye using some cutting-edge technology, and also hopefully sell to the rheumatologists where the eye might be a great organ to learn about aspects of immune function and autoimmunity that can't be done as easily in the joints or systemically. So hopefully this will get lots of conversations going. I'd love to be in contact with you, or please speak to your fellow ophthalmologists which you're embedded in. And I'll just start by saying that actually the eye is not this privileged organ that we used to think, you know, for the great Peter Medawar sort of put it in that box, and we've never moved out of it. But as the uveitis specialists here would tell you, it's capable of being profoundly damaged by a strong immune response. It's continuous with the CNS. So in fact, the part at the back, the retina, is a continuation of the central nervous system. But because the eye is naturally transparent, we can actually look in directly. And that's where some of the work I'm going to tell you about comes in, because we can see with exquisite detail down to single immune cells in a pressurized, untouched system. And we can certainly use that in a research basis in some of the mice models that we use, but also in the patients. And some of that's already beginning to happen in clinic. And to try and make a link for rheumatologists, remember the eye is also a joint. So the iris, the muscle at the front, has an enthesis, an insertion, and as many of you will know, with spondyloarthropathies is involved and inflamed in the same way. So I think that's becoming increasingly clear, and certainly we've been collaborating with rheumatologists in Oxford, and we're finding different populations that seem to match that in the peripheral joints in, for example, the iris and ciliary body insertion. So I think there is a lot more overlap than many of us would imagine. And from the eye side, I've always been passionate about immunology and the immune system, as many of us are. And I think we're increasingly realizing just how many more diseases are actually either initiated or worsened by immune system dysregulation. And so these are just a list of some of the more obvious, but now less obvious ocular diseases. And we're finding evidence that the immune system is deranged in all of them, but not just in diseases, but also in treatments. And some of the very promising treatments we have in the eye field, like gene therapy, stem cells, are now being hindered by immune responses to the very therapies. So we need to better understand what's going on in all these conditions. And I probably don't need to tell you, as certainly the rheumatologists, about why we can't just keep taking blood tests. And I know there's a big push to move to tissue level analysis of the immune response. So I think you're doing more synovial biopsies, for example, rather than blood tests alone. And in the same way, I think we're recognizing in the eye that blood tests and even ocular fluids are not enough to tell us really what's happening in the tissue, which is ultimately where the immune system works and where immune cells act. And so this is where I just wanted to point out to the eye team that we need to be able to directly image immune cells in our patients. And I think we really need that to better manage them, to preserve vision, and also to increase our understanding. And even though I think as ophthalmologists, we feel, maybe very lucky in that we can look in, for example, with a slit lamp and see immune cells sort of floating around the front of the eye, but they often don't reflect what's happening in, for example, the retina. And then we use a lot of indirect measures like leakage. So this is what we call an OCT scan of the macula, where we look for leakage in response to inflammation. So we're also looking at indirect measures of the inflammatory process. I just want to actually, can we get the lights down a bit? Because I'm going to show some videos. Thank you. And so I just want to orient those of you that aren't familiar with the eye into what it looks like, and I'm going to show some of our histology from the mice. So this is actually a whole mouse retina taken out. So you can see it's a half cup that would normally sit at the back of the eye, and this is just where we've stained it for the blood vessels with collagen 4 and imaged it with light sheet microscopy. So it's a sort of round ball. And you initially think certainly there are blood vessels there. And then when we do whole mounts, and this is beta-3 tubulin, that's not projecting perfectly, but you can see the whole sea of neurons in the inner, these are ganglion cells, and then they're forming together into nerve fiber bundles, and then they coalesce into the optic nerve, and then this ultimately goes through into the brain. And so most people, when we think of the retina, think of a complex neuronal structure. But actually, there I'm only showing you one marker on immunohistochemistry. But actually, it's also filled with tissue-resident macrophages. So here, the microglia in the retina are completely throughout the retina and surveilling it. So we need to remember that the whole space, that we normally consider just as neural tissue is, in fact, full of immune cells and a very rich immune environment, even in health. And so the bulk of my own work has been moving between sort of two species, modeling disease out in the experimental mouse models, and then also working in the clinic with patients with uveitis. And so this is really sort of the framework, and it's all very much linked around imaging technologies. And that would either be in living animals, so in vivo, sort of I'll show you some intravitreal imaging, but also doing a lot of histology now. And so I was in the US working with Ron Germain's lab for a year and a half. And I'm going to show you some of the techniques we've developed in the spatial biology field for better understanding the histology of what's going on. And the reason I show this is that it's a continuum that, for example, in patients, we unfortunately, like the joints, we cannot easily biopsy the retina without causing a lot of destruction and risk to vision. But we can biopsy our mice. And in the same way that, we can do some imaging while the animals or the patients are alive, it's much harder to get histology. But having the two and putting them together is where we're going to learn the most. So I'm going to start with talking about some histological techniques. And here is a human eye mid-dissection. So What we're looking at here is we've actually taken the white of the eye off. So if not familiar, here's the front of the eye with the lens and the iris. And then we've taken the sclera, so the white of the eye. And what you're seeing, that brown layer, is what we call the choroid and uvea. So this is the part where a lot of the immune responses occur and where the term of the subspecialty uveitis comes from. And these are eyes that we're able to get through the National Health Service in the UK. And we're very lucky in that we have a very joined up system. So almost all eyes that are given for organ donation or corneal transplantation, they normally throw the rest of the eye away. But we can actually now access them for research, and that's where we've been getting our tissue. And we can link it back to medical records to then find out what the patients had. So this has been a great resource. So what we do is when we receive eyes, we would prepare them like this. And then there are other elements we do. So this is a lovely picture on the left of a cataract, or the edge of a lens with a cataract, and then the ciliary body looking from behind and inside the eye. I always think that's very beautiful. But the part of the eye I'm most interested in is the retina, so that neuronal tissue. And incredibly, when you dissect the eyes post-mortem, there's still blood in a lot of the blood vessels. And you can see, for example, here, the yellow spots or the macula lutea, which is the very central part of the vision that many of the ophthalmologists will be familiar with. And so even just looking at that, we can get some information. And in the same way that we have those blood vessels there, we can actually use cutting edge technology in the eye clinics to image those vessels as well, either through fluorescein angiography on the right, or now through other non-invasive technologies like OCT angiography. But what we're missing in all of these is we're only seeing one element, and that's the point I want to make, that without being able to really fully phenotype the tissue, we're missing the full picture. And so we can also then do immunohistochemistry. So this is post-mortem human retina from a healthy donor. And what we're showing here is hopefully in green is the retinal arteries. We've then got in magenta the astrocytes, orange are the microglia, and then the capillaries and veins in cyan. And really just with 4 markers alone, you can really see how rich the tissue is. And this is just one tissue, let alone thinking about joints or many other organs across the system. And so it was sort of these images and being able to start using these tissues that got me very excited in histology and how we can develop in this space. And for example, we can then look at the tissue. So this is an example from a patient with diabetic retinopathy. So they have severe diabetes, and it doesn't look too dissimilar from what we see in the eye clinic when we look at the back of the eye. We see hemorrhages and we see abnormal changes, what we sometimes call beading to the blood vessels. But the difference here is this is a post-mortem donor. But what we can do is we can actually correlate this sort of bright field imaging with immunohistochemistry to then really begin to correlate the features we might see in our living patients in clinic with what might actually be going on at a cellular level that we'd otherwise miss. So what I think is particularly interesting here is there are these big dark patches where we're missing capillaries in the blue, which is well known from diabetes. But what we can now see in the purple or magenta are the astrocytes, and where are they clustering? And then in red, where are the microglia clustering? And I'll just zoom into this one area. I think the ophthalmologist will find this interesting. So this is what we call the microaneurysm, the dilatation out of one of the capillaries. And it's a well-known feature in diabetic retinopathy. But by taking the bright field image, we can actually correlate the two and see that there's blood still in this microaneurysm in the top right. And that seems to just be there with a blood vessel without any microglia around it. So IBA1 is a marker of the tissue-resident macrophages of the retina. Whereas the what looks to be the microaneurysm below in the bottom left doesn't seem to have blood anymore in it and appears to be fully encased in these microglia. So even just with these 4 markers in one tissue, we could begin to get insights on what might be happening at a cellular level in some of our patients, and then also how to better interpret the features we see in living patients on their color photography. But all I've shown you so far is just 3-4 colors, and I think many of you that will be doing research are familiar with immunohistochemistry. But we need to move beyond the sort of existing technologies we have. And we've gone from the beautiful drawings with CAML through to H&E, which is probably the workhorse in most pathology departments today. In many of the labs, those of you may be using immunohistochemistry, so using specific antibodies to one protein that you can fluorescently label. Normally, that's only 3 to 4 colors, and you can label, for example, here, different populations such as amacrine cells in the retina. But what I was interested to do was to try and see if we could push this technology forward. And we published a paper now a while back when I was in Ron Germain's lab. And there are now commercial solutions that are coming into this dimension. But this is great because it's an open source technology that you can use with your existing antibodies. And for us, that was particularly important working in the retinal field because many of our markers and cell types are rare and not invested in heavily by many of the companies. Essentially, what this allows us to do is stain on the same piece of tissue, a single section, upwards of 60 protein markers on the same tissue, unlike all the other ones in the conventional approaches, which just do 2 or 3. And there are different ways of doing this, but essentially what we do is we stain the tissue, image it on a microscope, and then we bleach the fluorophores. So we inactivate the fluorescence, and then go back and stain it again with another set of antibodies, go round and round, and then computationally align them. And what that allows us to get are these sort of beautiful images. So this is actually a human lymph node from the paper. And what we're showing you in each of these small panels is a follicle. And we're showing just 3 colors in each. But actually, all of these 60 markers are there for the same tissue at the same section at the same time. It's just impossible to display the richness of that data in this realm. And so while we've been getting a lot more information through the use of technologies like single-cell RNA-seq, we're now getting the spatial information as well. So this is a really exciting move towards that. And here's human retina. So we've similarly been able to deploy the same approach now in the lab and really in the retina be able to identify almost every main cell type by their protein signatures. So here I'm just flicking through the sort of, I think about 18 here, delineating a large blood vessel, all the different neurons, the different structural components. We'd be missing all of these elements if we didn't have this level of detail. And it's easy enough in this example where it's a healthy eye, but when disease occurs, it's even more unpredictable and chaotic. And so use of this level of depth of interrogation is even more critical. And we've been able to contribute this to many other ongoing initiatives. Many you may be familiar with the Human Cell Atlas, but we've been contributors to the HubMap consortium. And you'll see there are many people around the world now coming together to map the entire, sort of protein repertoire spatially of the human body. So this is the HubMap's now released several versions, and we were able to contribute this retina to it. And we're also very much about open source sharing. So if you're interested in this technology, we have a knowledge hub which we run where you can easily set this up. So any lab can do this. There's nothing special, no expensive technologies. So the commercial equivalents of this, you often have to buy their machines, which I think some of them started $1 million for the machine for getting the antibodies, whereas this you can use your existing microscopes and existing antibodies. And we're trying to make it as easy for everyone to uptake. But doing it manually, which is what we started with, takes a lot of time. I'm certainly conscious that for my students, they're spending a whole week doing this. So we've also made moves towards automating this process with microfluidic systems. So now the students can work during the day and the robot can work all through the night. So I think there's new challenges by having this new technology, but we need to accelerate that. And so those are going on in that space. And here's our sort of setup in London. So again, it's much cheaper if you want to get into this field than the commercial solutions. But a section, as I've shown you all before, just one thin slice misses a lot of the picture. And so this is some mouse retina, but I think it makes a nice example that here we've just got one thin section. But you might then miss the 3D structure of what's going on if you only had a single section, or even if you lined them up. So what we also need to move into, in the sphere we need to move into, is probably 3-dimensional and whole tissue imaging. And again, we've been able to do this, to about 3, 4, 5, markers at the same time, and using different sort of tissue cloning approaches. So here's the optic disc, and in the purple, you can see the distribution of a cell type called the amacrine cells, which you might miss that topology if you didn't have 3-dimensional imaging. And my group's been working with several other labs to try and get this so that we can build up the number of markers. And so this is human retina, and we're quite pleased that on the same section, by applying a similar technique, we're able to highlight the different structures and build up the numbers. So we're now up to nearly 16, and these are the microglia here. I just wanted to put some I know that Ines and some of you are interested in the gut, so this was with Andrea Radke, but this is human jejunum, a beautiful villi, which I just think is so spectacular. And here we've got several markers from 2 rounds, and then if I move the EpiCam, you can see the T cells in cyan all clustered around the villi, and really just to show this is in 3D. And so hopefully this work will be published towards the end of the year. It's a different approach we embed in agarose and then can do sort of 300 microns, but then push the number of markers up. So this one was an early one I did back when I was in the lab, but we're now probably up to about 20 to 25 markers in 3-dimensional tissue. So I guess what I just wanted to give a flavor of there is how on the histology side, we're really trying to innovate, increase our understanding, and then use that to map back onto what we're seeing in mice and patients when they're alive. And so I'm now going to shift slightly, and hopefully if you just bear in mind with all that histology being there to give us a foundation of what cells might be there, where they might be, let's now go into living mice first and then into patients. And so those of you that have not worked in animal ophthalmology, we actually can do almost every technique that we can do in the eye clinic on mice. And so back in London at Moorfields, we have what we call the mouse Moorfields, a whole laboratory where we have almost all the same instruments. And so this is an example of a mouse that we can just anesthetize. We use a different device. This one makes contact, but it's non-invasive. There's no surgery required. We just dilate the pupils, and then we can take images such as these. So just color photos, which look very similar to the images that we get when we examine patients in clinic, except the mouse looks slightly different. The optic nerve, if you're not familiar, which is in the center, is right in the center of the mouse eye, and you have radiating blood vessels. But things that we can do in the mice that we can't do in patients include inducing disease. And so this is a model of uveitis that we use called EAU. It's very similar to those of you that might be familiar with the MS mouse model of EAE. We essentially vaccinate the mouse against its retinal proteins, and then we get this progressive infiltration of immune cells and a vasculitis, which eventually then we get these infiltrates. So it models some aspects of human disease. And we're beginning to apply the same kind of high-dimensional imaging data to the mouse. So I've shown you the ibex with multiple markers, and this is very early data. But if you think about how many markers you need for your flow cytometry, for example, to really identify which immune cells you're seeing, this becomes even more important when you're looking on sections. So really, for example, we're now with our histology able to get down to T cell subsets with FOXP3 and raw gamma T staining, for example. And just looking at this section of the retina here, hopefully you can just see when it's infiltrated with immune cells, just how complicated and diverse it gets. And then if you even start to think about how many markers you might need just to define T cell lineage, what about, macrophages, for example? And so I think these techniques are going to be really helpful in autoimmune models as well. And the mice are never perfect models, we know this, of human disease, but they certainly share some similarities. So here's a perfectly curated image of a mouse eye, and then a patient with a type of uveitis called birdshot. So you can see there are some similarities. And then linking back to this imaging and histology, we can do these sort of cross-sectional scans called OCT, which you may have seen before, that are essentially a form of ultrasound using infrared light or near infrared light. And so we can actually then see these sort of little spots, these infiltrates of what we know from histology are probably immune cells on these scans. And these are the same scans we can do on our patients. But in the mice, we can then remove the tissue and then actually stain in 3D to see and show that these clusters are in fact collections of macrophages, microglia, and a few T cells. So the mouse is really useful for this direct correlative imaging, and we're using that now to better understand some of the observations we see with the histology. But we can do a lot more with the mice, and one of the things that we're really focusing on at the moment is immune cell tracking. And so we can do adoptive transfer, which I'm sure many of you are familiar with. You can essentially take pathogenic T cells from one mouse, for example, a mouse with a fluorescent tagin, like green fluorescent protein, and then inject those cells into the tailbone of a healthy mouse and it rapidly induce disease. So this is an example where we've looked at the back of the mouse eye after we've transferred those T cells. So all the T cells are fluorescently labeled in green. And so when we then look at the tissue and are able to turn that fluorescent reporter on, we can see that the tissue that is actually full of highly active T cells moving around, that if you just had the color sort of image alone, you would miss. And that's exactly the problem we have in clinic with our patients. We see these, we get sort of this image, just a sort of haze, and we have to interpret in our patients whether there's immune cells there or not. In the mice, we can do that because we can genetically engineer them. So then the question's always been, can we do something like this in our patients? Because then we could really, for example, count these, give them steroids, or maybe give them something more elegant like a biologic, and see if they're a responder or not, and actually know in the tissue whether the immune cells have depleted or they've stopped moving, and use that as a much better biomarker than what we currently have. And so we did some work quite a while ago that we're still actively pursuing. And this was using a dye that's used in the eye clinic. It's a crash. A dye in the eye clinic that we use all the time. Just reload that. Called indocyanine green, which is safe, has been used for nearly 60 years. And we found that if you give it as a depot, it actually macrophages and neutrophils ingest the dye, and it accumulates in endosomes, as you can see that. And we're then able to image them non-invasively in the mouse. Let's transfer time. Need to skip that one slide, see if there's something about that. Good job with lots of videos. See if I might have to run it without going full screen. Is it the memory of its own? Which ones? Oh, I think down to 30, a bit more, 30, a bit more, 43. 43. This one, yeah. Let's see.
Desde que ele se liga à
Yeah, we did. Go to 46. Yeah, that's right. Okay, maybe that's that slide. Let's not move. So essentially, we then looked at patients in clinic who are already having the ICG dye that we know is gobbled by the macrophages, and then imaged them non-invasively on some of our existing systems. And so we did see something quite interesting in that I've circled here. These are little dots. They're about the right size for immune cells, about 15 to 20 microns. But you can see the resolution and the magnification is not really good enough to be certain about that. And so we think this is really promising, but we needed a better way of zooming in, getting more magnification. And so that's what took me off to the University of Rochester, who are incredible engineers. It's sort of the home of the Kodak Photography Corporation in North America. And I work with them on this technique called adaptive optics, which is an optical technique that's now being sort of very cutting edge pioneered for correcting for higher order optical aberrations. So essentially what happens is normally the light when it comes into the eye is affected and disrupted by things like the tears and essentially defocused. But we can actually use this warping mirror called a deformable mirror that can correct in real time these moment-to-moment imperfections, making a correction of over 4 a second, essentially to get the sharpest and most incredible resolution in a living eye that's possible. And so this has been used in the past to image individual photoreceptors, but it's really only the very beginnings of coming into clinic and very much a research tool. So as I showed you, it's this sort of big optical table. We had to work with engineers to build it. But what it's allowed us to do is something pretty incredible. And so this is some work we published a few years ago. Essentially, all we did was use this adaptive optics correction to basically scatter shadows with infrared light. Completely given up this time. Scatter shadows with infrared light. And what we were able to see is that immune cells within the retina actually scatter the light and can be seen in relief against the transparent retina. So for the first time, we can get near single cell imaging of the retina. I wonder whether I'll keep it like this for the moment. So essentially what you're seeing here is the same mouse, this is a vein, the same mouse eye before any inflammation. You can see it's a smooth surface. We're in the inner retina here. And then after we give an injection of LPS, actually will cause inflammation, you can see all these cells here. Let's see if it will. Let me show you the next one, which is we were then able to take videos. We're able to then record essentially the cells moving. So we can non-invasively, I'm just wondering, do you think it's too large? Let me see if I delete the other slides and save. We're near the end.
Sorry.
That's okay. Deleting those. Or is it the memory that needs to be wiped? Computer might need. Is it the memory? Yep. But we don't have a connector. Zoom. You think? I've got my laptop, but no, it's HDMI only. The Zoom's running.
Can you connect with our account?
Oh, okay. I could try. Yeah. Do you have Wi-Fi? Yeah, I've got EduRoam. So that's okay. You have the meeting ID.
You have.
I've got Wi-Fi. Yeah. Do you have the meeting ID?
Like this, you connect to your
And share. Yeah.
yeah. That's great.
Okay. Yeah. And near the end.
Okay.
Got it? Working? Hey, hooray, we're back. That's the trouble with all this tech, too much technical difficulties. So essentially, when we image the mice non-invasively, we're able to see these moving cells. And what you're seeing on the left are macrophages and neutrophils moving in a live mouse. It's all we've done is give it a general pupils. And essentially what you can see here without any labeling, any genetic manipulation at all, just with this incredible optical correction and scattered infrared light, we can see neutrophils, and we used fluorescent reporters to confirm this. And then over on this side, in a healthy mouse, this is almost certainly a microglia on the surface. And hopefully what you could see is it's got its arms surveilling the microenvironment for damage. And the degree of resolution, these scale bars are 10 microns. So we're almost seeing subcellular structures within it. And this is all in a live animal, non-invasively. So this got us quite excited. We're seeing all sorts of cellular behaviors we've never seen before. We're not even sure what this is. I assume it's a macrophage or a neutrophil. But just the level of resolution we're able to get was pretty spectacular. And then we could look at different aspects of inflammation. So just to orient you here, here is a retinal vein, and in blue is where the vein is. The tissue around it is very inflamed, and the blood's actually flowing from the left to the right. So you'll see these streaks, and those individual streaks are single red blood cells. It's slow to zoom. And hopefully what you can see on the bottom, these are leukocytes rolling along the endothelium. So a behavior we all know about from the textbooks, that they roll, they stick to the endothelium, and then eventually come out into the tissue. But we're actually able to non-invasively image this in the mice and see them rolling. They're coming through on Zoom eventually. Just wait for it to catch up. And we were able to show in the paper that if we transgenically labeled these different cell types, we could confirm that they were in fact immune cells. And it's not coming through. Let's reshare. Dropped off from the Wi-Fi. Last few slides. Yeah, it dropped off. I think the internet cut out. It's back again. It dropped off. I just need the meeting ID again. Sorry. Yeah. I think the Wi-Fi dropped. Let me see. Yeah, it is, but it's low. Maybe let me just see if I can do the last few slides on nearly there. That's okay. It's just the last one. Let's just try and finish off with the... okay. Okay.
More challenging than labeling.
Makes the
Like
medicine easy.
pages in months.
Keep it like this. Oops. think that's actually, again, I think it's the sharing. But essentially, we can go back to the same location of the same mouse. We'll just wrap up because the last few slides. And that's the other exciting thing, because we're not doing surgery on the mice, exenterating, like mesentery or cremaster muscle that you might have seen in a lot of other approaches. We can find the exact same location of the organ and go back within hours to days to weeks to months of the same location of an immune response and track it longitudinally. So it's really exciting, and most people in their models, they can look at the early stages of inflammation, but it's very hard to then look at resolution or clearance or scarring or fibrosis. I think this is a great opportunity for us to be able to do that, because we can take the same eye, this is from the same eye of the same mouse throughout its immune response and resolution. Let's see if we can next. And we can also look at other aspects beyond the immune cells. So we can look at cardiovascular aspects. So we can actually, as you see in panel A, take our imaging beam, and then, those streaks that you saw in the video, each one of those is an individual red blood cell or erythrocyte. They're just moving so fast that the detector can't move quite as quickly. But you essentially get a line which we can actually then quantify in space-time and work out the exact velocity of that individual red blood cell. And we can just record in the same place of the mouse, for example. So what you're seeing along the bottom, we can just record that change in essentially velocity, and we can actually almost map out the cardiac cycle there. So you'll see the intervals where the velocity is accelerating. And so you could ask, this is probably the world's most expensive, pulse oximeter, for example. But actually, we can then directly measure in particular parts of the tissue and pick the exact vessel to see what might be happening in that region. And so here, for example, we can then do that same measurement in a coupled artery, capillary bed, and vein across an entire immune response again. So what you're seeing in these graphs here on the left is a velocity in the vein in blue, and then in the artery in red. And then basically the peak inflammation is around 6 hours in the first two, and then resolves. And what's really nice here is that again, in a conserved unit, we can show what we would expect. That is, the arteries increase the velocity of the blood flow to deliver more immune cells to the site, whereas the veins are incapable of doing that. However, if we then measure their diameter on these scans, you can see at the top that, that the veins dilate in response to inflammation, and that we can map out, whereas the arteries don't do that. And then overall, the net effect between those two changes, an increase in velocity and then a dilation of the vein in concert. And we can, in the retina there, they're sort of perfectly balanced interval units. We can then show that this perfect conservation of flow that increases and then decreases over the course of immune response. So at the moment, these are all pretty new technologies. But we're able to establish and confirm in living animals and sort of all the things that we've held to be true in terms of immune response. And the next steps are going to be really being able to attenuate or manipulate that and take it further and better understand the interplay of hemodynamics, neurovascular coupling, and the immune response. And we can do that all in living mice. But then the question is, can we do it in man and in patients? And the answer is we can. These same systems, these adaptive optic systems, we actually have or built for patients. We have to completely rebuild them to accommodate a different sized eye, but they exist, and people have been using them to image mainly actually the photoreceptors. But we've started a clinical study recently where we're now putting in the same sort of parameters that I've shown you in the mice, but into a human system. Because if we could do the same in patients, particularly patients, for example, with uveitis, we could really drive our understanding forward. And we've only just started this clinical study, which is sponsored by the Wellcome Trust. And here's actually a healthy volunteer, but hopefully what I can show you is at least where the red circles are. These are almost certainly sort of hylocytes of those resident microglia on the surface of the retina. So I think we know we're able to use the same approach. It should work in humans like it has in mice. We're still just at the very beginning. And actually some colleagues in Pittsburgh have actually been using a slightly different approach to get at this. But what's really exciting, shame probably won't play, but they're able to, this was in a patient with uveitis, and actually, again, hopefully you can see these structures, which we know are not normally there. So these are almost certainly macrophages, neutrophils. But what's really interesting here is this is a patient that's had 2 weeks of oral prednisolone, just showing how, you would expect, for example, that to be very effective at completely switching off an immune response, but the tissue is still full of cells. And I can't show you the video, but they're actually moving and still motile, so probably alive. So I think this will allow us to really do is then phenotype our patients to a much better degree, starting with patients with autoimmune disease in the eye, but eventually applying these techniques to many other eye diseases. And then we know that the eye can reflect many other conditions, multiple sclerosis. You may have heard of dementia, for example, being diagnosed through retinal imaging. So we'll be able to apply this to a whole range of other fields. And the other thing that would be quite exciting is then drug testing, predicting personalized responses to, for example, biologic agents, to steroids, because we can do this imaging because it's non-invasive, image the patients after they start treatment, and then see whether in that individual patient, their tissue has responded to the treatment by the depletion of immune cells. So I think it's an exciting time. It's still going to need quite a lot of technical optimization. We obviously show you our best images, but they take, sort of an hour of recording in the patients, and the amount of computational processing, so it's crashing all the computers, is quite large. But I think it's an exciting time, and we'll get there. So hopefully what I've just given you, an overview of is the sort of research we're doing, the sort of space we're working in between the mice and the patients, and then bringing you back to the original histological images. As you're seeing, all the in vivo images are very grayscale, and we can't be certain without a biopsy that those are definitely immune cells, or what immune cells they might be, or their state. So that's where using the mouse, where we can manipulate the system, training, for example, some algorithms, machine learning based on what we see. We're also going back to histology from patients, and that we do have to at least know what subtypes or types of immune cells we would expect to be there to try and then interpret the imaging. So for me, I find myself going round and round in circles between all these areas, and we're hoping in the future we'll be able to integrate them to really get tissue level immune cell imaging working in our patients. You finally got there. Just like to thank everyone and for the invitation here today, our funders and all my lab. Thank you. We got there, and thanks for putting up with the AV issues.
Thank you, wonderful talk, and thank you for overcoming those technological challenges, maybe more challenging than labeling the human than the mice retina. Anyone would love to make a question?
It's a specific question because my field of work is reproductive rheumatology. I wonder if you did any research in mice, considering pregnant mice studying the eye. Have you done any research in that field?
We've not, to say, but it's very interesting. Yeah,
but there are adaptive immune responses during pregnancy. So I was wondering if you did any research in pregnant mice.
Yeah, that's a great question. I have to say, I haven't looked at them just because, it's not been something that we thought of, but I think it could be really interesting. As you say, there's immune changes and also hemodynamic changes. So actually some of the cardiovascular measurements in the retina would be very interesting to see as well, as triggering some of the immune responses and seeing. So it's certainly something that could be done. Yeah.
Also, it's not my field, but there are animal models concerning, for instance, spondyloarthritis. I think it would be also good to study the eye in those specific arthritis animal models or spondyloarthritis animal models. Yeah,
that's a great thought. At least in the eye, most of the inflammation tends to, in those sort of models, tends to happen more towards the front of the eye. So there's certainly still a lot we can learn with the histology. In terms of the imaging we showed you, it works best really in the retina. But we do see some overspill and some interplay there. But yes, it could definitely be done.
Thank you, Professor Colin, for your amazing talk. Would it be possible to combine adaptive optics and in vivo fluorescence?
Yeah, absolutely. So yeah, that's a good point. So adaptive optics is essentially just a technique that can be combined with any other imaging modality. So we can do adaptive. What I've shown you here is we call adaptive optics scanning laser ophthalmoscopy, but we can also do adaptive optics OCT. You can increase your resolution, so then you can see individual cones. We've got a clinical trial starting in birdshot with a collaborator. They're doing something called optoretinography with adaptive optics, where you can see the individual cone photoreceptors, and in response to flashes of light, they shorten by 0.5 micron. And so you can measure that and essentially do an ERG using OCT scans. So yeah, it's a good point. And so that's exactly one of our plans with the fluorescence imaging. So potentially with the ICG dye labeling I was showing you is to then use an adaptive optic system to get the magnification and the resolution. So yeah, great point. And that's where we're going.
Thanks for your talk and for this amazing work.
Thank you.
Would you, two questions. One is more methodology. Do you anticipate that it's, could you reassemble these tissues on an in vitro level, like the human tissues, not the rat one, to study it as you study in the rat? Because here you have a really big advantage that we don't have, that the eyes are really in front of you and you can achieve, like the skin. And for us, it's a bit more difficult. Could you anticipate and try to reassemble the tissue and study it on that level? And the other question is more clinical. Would you see the possibility of looking on these imaging patterns and histology patterns to like find some categories of uveitis patients that you could stratify and even in terms of therapy?
Great, thank you. There's two really great questions. The first one, I think if I understand correctly, is re-imaging the same patients with the adaptive optics system, longitudinally, the same tissue region. Yes. So we can absolutely do that. Because the retina has very precise capillary structure, you can literally find your way, you navigate the map from the optic nerve out along the blood vessels. So you can be very confident you can get within a few microns of exactly the same location in the same patient. So yes, that's probably the most exciting part, because then we can image the exact locations, see where the sort of, the focus of inflammation is, and then go away, treat the patient, bring them back a month, a week, a day, a year later, and re-image the exact same location. So yes, we can definitely do that in patients as easily as we do in the mice. So that's very exciting, and where, as you say, it's harder to go back to a joint biopsy, for example. So yeah, that's a really great point. In terms of the histology, you're right, using it, I guess, as a biomarker to stratify, we definitely need to do that because in uveitis, we just don't have the numbers. They're quite rare diseases. We actually don't understand half of them. We're probably treating a lot of conditions that are inflammatory that are not inflammatory, or others that are inflammatory that we're missing. So we definitely need to do that. The problem we have is the lack of histology. Just the real problem for us is we can't biopsy. It's almost just not done because it leads to complete blindness. So we only biopsy when it's sort of already, sort of in the end stages. But not as much as used to be done. So it's actually quite rare these days. We've kind of got good enough to preserve eyes. So in a way, we're not accessing histology to get enough numbers to stratify. But we are lucky at Moorfields. We have a big archive of old eyes with patients with uveitis that we're now going back to and applying these techniques to look. And I hope that will give us information.
But I meant also with imaging patterns, not only histology.
Oh, sorry. Yes, completely. I mean, we don't know, right? So I'm hoping that we just don't know yet, but I'm hoping that's the case. Because we do see different patterns, as you've seen in the other images, of clumps or just uniform distribution. And so what we don't know, though, is if the immune cells are around that area or not. But exactly that, I think it's a new horizon, and I hope we'll be able to stratify and then understand in that way. We should certainly at least be able to work out, for example, our responders and non-responders. So for us, I think it's well known uveitis, only about 60% to anti-TNF. And trying to predict those, for example, from a biomarker like this would be really helpful.
Thank you.
Thank you.
Thank you for your excellent talk, very inspiring.
Thank you.
I would also like to know if you see any possibility of any of those technologies being able to see through the skin for a very superficial joint like hand joints.
Yeah, it's a great idea. So the that infrared one, I think, will not be able to penetrate deep enough. So you can do the skin, probably, get down to the dermis, but then just the way that works, I don't think you'll be able to get much further. I think where might be useful is the ICG dye, because it fluoresces sort of, at a slightly longer wavelength. It can shift up to around 1,000 nanometers. You might be able to do that. So maybe what we should yes do is when we're planning a trial where we're going to move to the next stage of that ICG labeling in patients. So then while we're imaging their eyes, maybe we should try and image their joint. With that, enough fluorescence might escape, as you say, from a superficial joint. So it could be exciting. That's a good idea.
And regarding the size of the equipments, is there any possibility of putting kind of a probe inside the joint? Because inside the joints, the synovial is fairly transparent. So if we could see it like that during an arthroscopy, like
Yeah, wow. So currently, the instruments are bigger than this table, so it's going to be a challenging approach. But I don't see why it's not impossible. So people are now using fiber optic coupled systems. So in theory, then you might be able to do something like that. But I'm not aware of anyone doing something like that at the minute in the eye field. But it's a great idea, and probably some of the concepts will translate. But it partly depends on the environment. I think we're very lucky that the retina is naturally transparent, so the immune cells sort of show up because they scatter the light. I don't know if that would be the same in the joint, but then the synovium is quite, flat and you might see it. So I think it's a great idea.
Thank you.
Thank you. Brilliant. Well, thank you all so much. Very happy to chat afterwards as well. And please reach out. I think Inesh has my contact details as well. Thank you so much.